So today my talk is going to be, I'm not running a lab, I'm not an independent researcher. I'm actually going to describe a suite of tools and biological findings that's been developed both by, me in the past. I like to call myself a retired bioinformatician these days, although I still code sometimes. Also tools developed by PacBio's own CompBio research group, and also our collaborators and customers. And it's going to straddle between informatics and also biological insights. But let me first set up the problem statement. We're heading into the multiomics era. But when you look at the multiomics assays, a lot of them contain fragmented and incomplete information. I'm talking about the fact that with short reads, you cannot see isoforms. With long reads, you can. With short reads, it's very difficult to resolve things like segmental duplications, tandem repeats, but with long reads, it's much easier. One slide about what HiFi is. It is PacBio's long-read sequencing offering. The reads for WGS would range from 15 to 20 kilobases and are highly accurate. HiFi has been used for assembling T to T genomes, folding transcriptomes, epigenomes. But with these new data sets, really the problem, and the one that I'm going to focus on in this talk, is the computational tools that are required. They're different. When you go from short reads to long reads, a lot of the assumptions and heuristics that were developed for the short read bioinformatics tool fails. So here's just one of the list of the problems and the tools that have been developed for long read in the DNA space. I'm just going to talk about two of them, but maybe we can just look at three of them in the year. For variant calling on SNPs and small indels, we've partnered with Google to create deep variant. For tandem repeats, there's Target, which I'm going to talk about. And for distinguishing segmental duplicated genes, I'm going to talk about paraphase. And in fact, this morning, our CompBio group has released MathBat for methylation calling, and you can find it on GitHub. The parallel to this in transcriptome is isoform classification, which has just been published in SCONTI-3. This is a collaboration that Anna Conessa's lab started. I was the main developer for SCONTI-2, and now they've taken it even better to SCONTI-3. But that's not the only tools that are there. I like to mention FLAIR, Talon as other alternatives to isoform classification other than SCONTI. Deep variant has been shown to be able to work for RNA-seq data as well. I'm going to talk a little bit about fusion detection, quantification, and visualization. So let me start with the DNA tools. First, let's talk about tandem repeat. Tandem repeats, there's more than 1 million of them in the genome, and a lot of them have clinical implications. They're associated with autism spectrum disorders, schizophrenia, and a variety of neurological diseases. But measuring tandem repeats is very hard because a lot of times these repeats there's hundreds of copies of GAA or pentanucleotide repeats. It's very difficult to resolve them with short reads. Not only you want to know the accurate repeat length because it is the length that often will infer pathogenicity, but you also want to know about the sequence composition. A lot of times these repeats are not pure, and whether they're pure or not also affects their pathogenicity. So Target is developed by Igor at PacBio's CompBio group, and it is used for tandem repeat genotyping and visualization. Here's an example of how Target is applied to an actual clinical disease case. So RFC1 repeat is polymorphic in length and sequence composition. The biallelic expansion consists of several different pentanucleotides, and the AAGGG motif is known to cause the canvas disease. So if you look at this particular sample, there's the two haplotypes. Below, you're seeing this AAAG being expanded, but this is actually not pathogenic. The pathogenic allele is actually on the above one, where you're seeing 5 different motifs being expanded. And in fact, it is the orange one, the AAGGG, that's the one that's pathogenic. I will later show you a publication that's looked at using Target to look at a variety of other diseases. Next, let's look at segmental duplications. Segmental duplications are very difficult, and yet they harbor a lot of genes. These segmental duplicated genes can be more than 99.9% similar. Paraphase is developed by Xiao Chen at PacBio's CompBio group, and what it does is noticing the fact that these segmental duplicated genes are very difficult to map, almost impossible to map in short reads, but even with long reads, it's still difficult to map. So what it does instead is extract these reads from these genes of interest just to that gene. So I'm going to show you an example in the next slide. Phase these genes into haplotypes and then annotate these haplotypes, because often it's these annotations that's going to tell you whether this person is going to be a carrier of a certain disease. This is the example of that. So SMN1 and SMN2 are segmental duplicated genes. The loss of SMN1 causes spinal muscular atrophy, which is a leading cause of infant death. If you look at this particular data set with the IGV screenshot, on the top, if you're using short reads, 84% of the reads are ambiguously mapped. If you use long reads, there's still a decent amount of misaligned reads. So instead, if you use paraphase, it can very clearly distinguish the haplotypes. It tells you that there's 2 copies of SNM1, 1 copy of SMN2 with a deletion in the middle, and then 1 copy of SMN2. So that's the two tools I'm going to talk about on the genomic side. Now I'll talk about RNA tools. But first, I like to visually show people just how different RNA-seq is in the short and long read space. If I show you this graph from the left to the right side and ask you, can you tell me how many isoforms there are? The left is what the short read RNA-seq world has been doing for years, right? First, we start with transcript assembly, then we do inferencing, and then we start just counting k-mers. But on the right-hand side, you're not doing any inferencing or assembly. What you see is what you get. Every read is a full-length isoform. This problem becomes even more difficult in the single-cell space because of the necessity to sequence and see the UMIs and the barcodes that tells you which transcript comes from which single cell. You're now limited, as the previous talk has shown, to a limited number of bases next to the poly A tail and the 5' and the other end of the fragment. This is not a problem with long reads because you are sequencing the entire single-cell cDNA. Now, in the following slides, I'm going to talk about the bioinformatics tools. You'll notice that most of them seem to focus on bulk RNA sequencing. It's not necessarily true. It's because the following problems, classification, variant calling, phasing, and differential analysis, once you solve them in the bulk space, I believe most of these tools translate quite well to the single-cell space. In fact, for SCON-T3 of isoform classification and filtering, this is now implemented as Pigeon in PacBio software SmartLink. We use them both for bulk and single cell. There is no difference in how you classify bulk versus single cell RNA. What SCON-T does is solve a problem that didn't exist until long reads came along, which is now I'm not just relying on GenCode or RefSeq. I have novel isoforms. What can I say about them? And I think I do give credit, Anna, so much credit for coming up with these nomenclature that I believe now people who work with long-read RNA-seq has widely adopted. To be able to say, here's a known transcript, here's a novel transcript, and this is why it's novel. It's using a novel junction, it's using a novel donor site, or if it's an incomplete splice match, which is the bane of everybody's existence because it's either degraded 5' ends or actually biologically meaningful, and we have great debates about how to deal with that. But the isoform classification and filtering is the first step to being able to work on all the important problems downstream. What is the function of the isoform? You can look at it from the protein domain. You can look at it with an association with other genes. And then you can do differential analysis on the isoform level. But interestingly, I would argue differential expression analysis isn't very different than gene level because they're all just statistical tests. Instead of using genes as features or exons as features, now you're using isoforms as features. What I think has been underappreciated and maybe somewhat cast doubt on for the years since I've worked at PacBio on long-read RNA is I don't believe these novel isoforms are real. It must be a library artifact or informatics artifact. I think the evidence is that a lot of these novel isoforms are real. It's just we've never seen them because we've been very driven by GenCode annotations, even though GenCode annotations have been expanding for years. Just to give you an example, as early as 2022, customers found that 2/3 of the breast cancer isoforms found in their breast cancer cell line were novel. Look, they were using Sconti's nomenclature back then. And then in 2023, looking at 10x single cell colorectal cancer samples, they also found 1/4 of the isoforms are novel. These novel isoforms are sometimes really important. They can be private or disease specific. For example, in this publication, they looked at ovarian cancer patients using 10x single cell data. They found that there is a novel cancer specific, but also patient specific fusion transcript that's shown here in one of the 3 ovarian samples. What's interesting is that when they looked at the matching short read data that was generated, it never showed up this fusion. It actually misclassified every read as being TESPA1. So when you did a differential expression test and says, what's the most distinguishing gene between the cancerous cells and the non-cancerous cells for the sample, the short read answer would be TESPA1, when the real answer should be it is the fusion transcript that's differentiating the cell types. Which leads me to talk about fusion tools. There's many fusion tools for long read out there already. I'm merely mentioning one that's developed by Zev Cronenberg and Roger Voden at PacBio's CompBio group called PB Fusion. You can find it on GitHub. Kind of the final topic I want to touch on before I move into looking at biological insights is variant calling. So a little walk down history. In 2020, me and a few collaborators at Cold Spring Harbor published what I think is back then probably the first GitHub repo, I'll just say GitHub repo, okay, on using PacBio ISOSeq data for doing allele-specific isoform expression. We didn't even do it on human back then. We did it on maize because my collaborator works on maize and sorghum. We showed that with long-read isoform data, you can not only call the variants, but you can also phase them and then show maternal and paternal specific isoform expressions. But it took another 3 years, maybe I'll blame the pandemic, for me and my collaborators to publish another paper, this time looking at finally human data. We looked at WTC11 and Jurkat cell lines, where we, instead of writing new tools, we benchmarked D-variant versus CLAIR3 and GATK for SNP calling and variant calling. Now, what we showed was that D-variant with some modification. And the main modification is to take into account of the splicing. Because remember, D-variant was trained on genomic data, so it doesn't understand splicing. So if you don't do some pre-processing, it essentially treats all splicing as, oh my God, there's a giant variant in there. It's not a variant splicing. But once you take into account of splicing, D-variant actually works quite well. Now, fast forward to 2024, just within the last 3 months, I've seen 3, I named only 2 here, actually 3 long-read RNA-seq phasing tools come out. LongCallR is on GitHub only. I don't think I've seen a preprint yet. IsoLaser just came out, I think, 2 weeks ago. It is fascinating. It analyzed the ENCODE for consortium ISOSeq data and showed it was able to classify cis versus trans splicing, which I'm not showing here. It's fascinating stuff in the preprint, where they show that immune genes, such as HLA genes, are highly enriched in individual-specific splicing, and that different alleles of HLA genes actually have different preferential splicing patterns. These are things that you can only do when you have large amounts of long-read RNA-seq data, and I do think we're starting to get there. So with the last 5 minutes, I'm trying to go fast, I'm definitely ambitious in the number of slides I have. Questions on depth. How many is enough? To revisit the 30x genome paradigm, if you're in the genomic space, maybe not, is that this was based on a 2008 paper based on short read. How short? 2 by 35 base pairs. So we revisit the problem. How much do you actually need to do to get the answers you want? If you look at PacBio HiFi data, it shows that you can get similar recall on SNPs at 9x and indels at 20x. For structural variations, it's performing pretty well at 15x across different tools. With short read, structural variation is just very difficult, so you can see why it's totally at the bottom. However, de novo assembly will always require higher depth than alignment-based variant calling. So we see that by looking at the number of contigs, which less contigs is better, and higher M50, which is the orange bar, is better, that you probably will still need somewhere between 30 to 35x coverage. So this is the conclusion so far for genomic death, right? So somewhere between 15 to 20x for capturing variants and 30 to 35x for genome assembly. But what about the transcriptome? So I'm actually a product manager these days. This is what I've been working on, is we released a new kit called Kinnex for RNA sequencing. There's actually for single cell, for bulk, and for 16S. But diving directly into the data that I've worked with my collaborators on, we compared Kinnex with short read data on a differentiated WTC11 cell line from day 0 iPSC to day 5, differentiated into endothelial cells. Just to show you some of what the data looks like, is the reads are not created all equal. Just in genome assembly, the long versus short reads are not the same length, because when we compare the 5-day data and the replicates at the number of quote-unquote reads, they look about the same. But actually, if you look at the number of bases, there's 10 times more bases in the Kinnex data. Why is that? Because the Illumina data is 2 by 150, and the Kinnex is anywhere between 1 to 10 kilobases. It's the entire transcript. However, if we look at simply saying, can I get a gene? Can I get an isoform? Short reads at the same depth will do better because of the inferencing and the reliance on using references, whereas PacBio is trying to look at the entire transcript. However, the difference, I would argue, at the gene level is negligible. If you look at the 2 lines of genes, we're not seeing any difference, but you will be able to call at the same depth using inferencing tools slightly more transcripts than PacBio. Now, when we look at differential expression markers, to be honest, maybe this is too simple of a problem. We don't see a difference. So like at 5 million reads, we can easily call differentially expressed endothelial markers at 5 million reads for PacBio. So I would say the sequencing depth for long-read RNA-seq, at least to me, is just work in progress, because we've just started to be able to generate hundreds of millions of reads this year with the launch of the Kinnex data. Let's see if I can get through the publication highlights. So what I wanted to showcase is what is all the exciting biology that's coming out of this. So one is the analysis of thousands of HiFi genomes to find a novel flanking variant that is actually not the reference. So we keep saying the reference is the reference, but it's not. In this case, the variant is actually pathogenic for this repeat expansion disease. And it actually showed with analysis of these genomes that the actual common variant CFV is actually ancestral and actually confers tandem stability. And down here, I'm showing like, what are all the tools you need to call this? You need many tools: target, devariant, PBSV, and HiPhase. Switching to RNA, I'm really excited from this new preprint that just came out from UCL, where they're looking at Parkinson's disease using ISOSeq. So if you're wondering UTRs are ever good for anything, well, it turns out SNCA, most of the diversity is actually in the UTR region, where they're actually able to design, based on these novel annotations, antisets oligonucleotides, and then actually show certain modifications on the ASOs actually reverse Parkinson's pathology. So the tools used here are ISOSeq, Sconti, and GG transcript for visualization. Okay, 2 more slides. So if we look at novel segment duplications, this is looking across hundreds of HiFi genomes. They're able to discover 7,000 segmental duplicated genes, and the ISOSeq data supports like some of these are novel protein coding. And finally, we talk about multiomics. You have to combine the genome, the methylome, the epigenome, and the transcriptome. This is a wonderful preprint that I really encourage you to read if you want to see how to throw a kitchen sink at one rare disease patient, where they're able to say, I can see the genomic structural variation and it causes a fusion transcript because the ISOSeq data showed it, but then I can also look at the chromatin accessibility to show the enhancer activities, and I can look at the methylation pattern to look at, particular loci silencing. I'm not going to go into the details, but just to say, this is what you have when you get so much different data and the amount of tools that go into solving one problem. So I hope I've convinced you that we are moving on from traditional multiomics to synchronized multiomics. And here's my summary. And if you want to learn more, please visit PacBio. I'm also active on social media. Looking forward to talking to the rest of you for the rest of this conference.
Thank you. Any quick questions? Can you go to the mic?
1, 2, 3. Yeah. Hello there. Thank you for the presentation. And I have a question. I'm currently working with huge amount of PacBio data. We have a collaborator from UCSF. They generated millions of long reads, and they have like matched short reads from the cDNA. And actually, the correlation is so low. It's like 0.3 something. Do you have any idea why it's the case? Thank you.
So they're both RNA-seq data.
Yes, yes.
Yeah, that would be very highly unusual. I didn't include this slide, but we've actually shown that it should at least be 0.8 or 0.9. So we can talk about what went wrong.
Okay,
Data
sure.
set.
So you're getting high correlation.
Yeah, we get high correlation in the WTC11 data set. So maybe we'll talk about like what could have gone wrong. I would be very surprised.
Hi, thanks for the interesting talk. So I'm wondering if 100% of the long reads are full-length molecules. So if not, like, do you have estimation, like how many are not full-length?
That's just what is exactly Anna's tool Scanti tries to tell you, right? Not only, there's a lot of other orthogonal data you can, like, what do you mean full length, right? Is it full length to the 5' cap? If we get to that point, then you have to talk about whether you're generating the full length cDNA. Because I think now the problem is less about sequencing the reads, because we can sequence. If you can make a 30 kb cDNA, we can sequence it. The problem now is the chemistry to be able to generate a full-length cDNA. So what we see a lot is three-prong bias, is that for really long transcripts, I'm just going to mention Titan. I have yet to see a 30 kb cDNA of Titan being sequenced on PacBio. I've only seen, maybe 10 kbs of Titan fragmented along the gene from, both the poly A tail and the DT priming of, intronic regions.